Do not get off the train because AP Bio is continuing. Up next, we're doing cell membranes, we're doing cell signaling, cell cycle. Don't forget, if you really want to get the 5, you got to dig deep in your review book. You got to answer the questions. You got to make some flashcards. You got to go a little further. This is going to be a good overview after you do that stuff. Okay, signal transduction. This is a topic that is pretty important to learn about. So the basic pathway of signal transduction is in 3 steps. You have the reception, the transduction, and the response. Number 1, the cell is going to receive a signal from a molecule known as a ligand. Ligand is the molecule that is going to give the signal to a cell. The part of the system that the ligand fits into is called the receptor. The ligand fits into the receptor, G-protein coupled receptor, and a tyrosine kinase receptor. Okay, both the G-protein coupled receptor and the tyrosine kinase receptors are associated with receptors on the cell surface. And what they do is they are going to elicit a response for something to happen in the cell, and then in the case of like a G-protein receptor, it will ultimately activate a secondary messenger. This is a situation in which the signaling molecule does not pass through the membrane. In this case, the signaling molecule, it is not going to be soluble in lipids. It is going to be hydrophilic. It may be large. It is not going to be able to cross this membrane. The second type of signal transduction process I want to go over is where it does cross the membrane. And in this case, the receptor is actually on the inside, and the ligand actually can go through the cell membrane. In this case, it has to be lipid soluble, and there's a class of molecules within the lipids that's considered a lipid. It's called a steroid. So a steroid hormone will cross the cell membrane barrier. Steroid hormone is hydrophobic. It will readily go across the membrane. It will bind to the hormone receptor intracellularly. That means that it's on the inside of the cell, in the cytoplasm. It will actually form a hormone receptor complex. This hormone receptor complex will cross the nuclear membrane and most likely will trigger specific genes to be turned on. Now, one example that comes up is going to be sex hormones. So, for example, the sex hormone testosterone is a steroid hormone. It will cross the cell membrane, form the hormone receptor complex, cross the barrier into the nucleus, and cause DNA to be transcribed into messenger RNA in order to make many growth factor proteins. Okay? All right, on to the cell membrane. Cell membrane is a bilayer made of phospholipids. If we look at this diagram right here, the outside of the cell and the inside of the cell both have the same component. These are the hydrophilic heads made of the phosphate groups of the phospholipid. And on the inside, we have the hydrophobic tails made of 2 fatty acids. This brings us to the fluid mosaic model is the model to describe the cell membrane. Why do they call it a fluid mosaic model? Well, the fluid part is just like water. Think about it as like an ocean moving around. So the fluid part is going to be the phospholipids. The mosaic is just like you would see in art. Mosaic means different pieces of stone or glass or different things put together to create a bigger picture of something. Well, you have all of these proteins and you have all of these things associated with it besides the phospholipids. It's actually considered a mosaic. So that's where they get the fluid mosaic model. Now these proteins in there, they're all different types. Some of them are channel proteins that allow stuff to flow through like a channel. Some of these channels can open and close, like we saw with the permeability of sodium and potassium ions when we talked about the action potential in nerve cells. Some proteins embedded in there have these carbohydrate units on here. These are going to be considered glycoproteins. Glycoproteins and glycolipids are considered ID tags for the cell. Up is the topic of osmosis. Osmosis is going to be the movement of water across a membrane. It's a classic example of taking different cells and putting them in different salt solutions. This is going to cause the cells to either shrink or burst or remain the same. Well, hypertonic, isotonic, and hypotonic all refer to the osmotic concentration of water. In other words, how much salt or sugar is in the water. They all have the word tonic. Tonic refers to dissolved substances. For example, know what tonic water is. It's not just water. It is something that people drink that is going to have flavor in it. So there's something in it. Hyper means more dissolved substances. Hypo means less dissolved substances. And iso means the same. Now, everything is relative. When we say something is in a hypertonic solution, that means it's relative to the other side of the membrane. So you can't just have a beaker of water and say, this is hypotonic. You can't say that. What you have to do is you're comparing it to another side of the membrane. So if this cell is in a hypertonic solution, that means the outside of the cell is hypertonic. That means it has less salt. Water actually moves from high concentration to low concentration. It moves from an area of more water potential to less water potential. It's going to come out. Water will come out. It will cause it to shrink and shrivel up. The opposite, hypotonic, is pure water. The term very commonly used for pure water is DH2O. D stands for either distilled or deionized, which means pure, nothing in it. So if a cell is put in distilled or deionized water, what will happen is that the water will travel into the cell. Isotonic is the same. That means the net movement in and out of the cell will be equal. Animal cells, particularly like blood cells, prefer to be in an isotonic environment. Plant cells, on the other hand, prefer to be in a hypotonic environment because with plant cells have a central vacuole. And when water needs to be forced in so it can become turgid. Turgid means that it starts to swell a little bit, but the cell walls keep it from bursting. So that's great to start the process of water going in the plant to either conserve the water and store it in the vacuoles, or to start the process of transpiration, because in order to get water up a tree, here's your tree, it needs a little pressure. They call that root pressure when water actually goes in due to the osmotic pressure. We can actually calculate water potential. Now, when we review the math, we're going to have a math section review. We're going to go into some examples. But for now, we're just going to talk about theoretically what water potential is. So water potential means literally the potential for water to leave an area and move across the other side of the membrane. So if we look at a membrane and we have molecules of salt, NaCl, and let's just say that it is 0.2 molar salt. If on the other side it has no salt and it's 0 molar, which side has a higher water potential? Well, before you even think about that, you probably will know from what we just discussed, which way does water move? Water will move from an area of low salt to high salt. It will be going from a region that is hypotonic to hypertonic. So what does this mean in terms of water potential? Well, water has the greater potential to move from the hypotonic side. So that means that the water potential, which is symbolized by the symbol psi, is going to be greater. It also means that the water potential on the hypertonic is going to be less. Now, there's two factors that contribute to the water potential. It is going to be the pressure potential and the solute potential. Both are going to contribute to the water potential because if you're dealing with just an open system and you're talking about at sea level, we can negate the pressure potential. And what's going to happen is that only comes into a situation when the cell wall is actually pushing back. So to calculate the water potential, you simply have a system where it's not discussing the cell wall pushing back. It's an open system, such as a beaker of water or in the surrounding fluid of a cell. All you have to do is calculate the solute potential. That can be found on your reference table for AP Bio. Okay, so we'll do practice problems with that when we do the actual math unit. Pure water, 0 molar, the solute potential of that is going to be 0. Therefore, the water potential is going to be 0. And when we talk about dissolved salts or sugars, it actually has a negative water potential. So if you think about it, a negative is actually less than 0. That is why a negative water potential is going to be less than 0. In these problems, once we get into it, you're going to see that the highest you really get in water potential is 0. You're really not going to get greater than 0. It's as high as you get is pure water. Any higher than 0 is going to be some factor with the pressure potential where something's actually maybe squeezing, like maybe a contractile vacuole or something like that. In a paramecium is actually squeezing to reverse the water movement. Active transport versus diffusion, which is on the order of passive transport. Active transport requires energy. Active transport very often are going to be by the nature of pumps in a cell membrane. The molecules they often pump are like sodium and potassium is a great example. Also, hydrogen is a very famous example of hydrogen. In the case of the sodium-potassium pump, this is what sets up the resting potential in a neuron. In the case of the hydrogen ion pump, this is what is going to occur in both cell respiration and photosynthesis in the electron transport chain, in which it needs to utilize the energy from the electrons moving down the chain to set up a high concentration of hydrogen ions so it will later flow through cell membrane by diffusion. Now, as far as diffusion is concerned, diffusion requires no energy, no ATP. It's going to move from a high to low concentration until equilibrium is reached. A lot of people think they say from high to low, and it doesn't switch spots. It goes until it's equal on both sides. There's two types of diffusion. There's simple diffusion, and there's also facilitated diffusion. Facilitated diffusion is where it needs the help of a carrier molecule, or such as a transport protein across the membrane. Simple diffusion is where it will just go from high to low and does not need the help of any kind of transport. All right, up next is the cell cycle. Cell cycle is going to have two main categories. It is going to have the M phase, or the mitotic cell division, and the rest of it is interphase. So I just highlighted the M phase so you can see it different than interphase. Interphase can be broken down into three parts: the G1, the S, and the G2. G1 is named because it has a general cell growth right after the mitotic division. The cells may be a little bit smaller, and they have to generally grow. They have to produce some more organelles, and they basically have to mature. After the G1 phase, they may not be ready to divide just yet. The cell cycle is all about controlling when and if the cell ever divides. Cells that divide uncontrollably is cancer. Controlling the cell cycle is key in preventing cancer. So cells that do not divide are right after the G1 phase, are in the G0 phase, which is known as the resting. The S phase is the part of interphase where all of the DNA replication is going on. So the S phase actually doubles the DNA. Up next is the G2 phase. The G2 phase is going to exhibit additional growth, organelle duplication, as well as enzyme production, really getting ready for the mitotic cell.